On the left, a pediatric brain scan where tissue fails to grow, resulting in continuous seizures. On the right, an adult's pupil constricts to a pinpoint, the first sign of terminal respiratory paralysis. These two events occur at opposite ends of the human experience, but they are driven by the failure of a single microscopic molecule, L-serine. It is technically classified as a non-essential amino acid, a label that suggests the body can easily handle its own supply. That label hides its actual role. Serine provides the physical insulation for the brain's wiring and functions as the specific co-agonist required to open the receptors that form human memories. To understand why these crises happen, we have to deconstruct how the disruption of this one reactive molecule stops the assembly of the nervous system and explore the laboratory pathology used to detect the damage. Serine links basic carbon nutrition directly to neurological drive. Its unique chemical configuration makes it an essential structural tool, but that same configuration creates a vulnerability that can be exploited by some of the most toxic substances ever engineered. Structurally, serine features a reactive hydroxyl hook on a 3-carbon frame. While most human proteins use left-handed molecules, the nervous system breaks this rule, expending energy to break and reassemble L-serine into a perfect mirror image, D-serine. This mirroring happens inside astrocytes, star-shaped support cells that surround our neurons. Because neurons cannot manufacture their own serine, they rely on this astrocyte neuronal shuttle to supply the mirrored molecules directly to their synapses. Without that supply, the NMDA receptor, the structure responsible for neuroplasticity, cannot function. While the neurotransmitter glutamate binds to the receptor, the channel will not open to allow the calcium influx needed for learning unless D-serine is simultaneously docked at its specific site. This dependency extends beyond our own cells. Specific strains of bacteria in the human gut also express the enzymes needed to manufacture D-serine. This bacterial supply is reabsorbed into the bloodstream, where it enters the same neurological networks we use to focus and form memories. The ability to encode a long-term memory is physically tied to the constant, energy-intensive mirroring of this single microscopic structure. When this manufacturing line fails at birth, the result is neurolaxova syndrome. A genetic mutation in the PHGDH enzyme disables the astrocyte's ability to synthesize serine, starving the developing brain of its primary structural resource. Without serine, the brain cannot build sphingolipids, the fats that make up the myelin insulation for our nerves. Uninsulated neurons misfire continuously, leading to the microcephaly and intractable seizures seen in Act 1. Inside the enzyme acetylcholinesterase, serine 200 acts as a chemical blade, but weaponized organophosphates mimic normal neurotransmitters, flying into the enzyme's pocket to form a permanent covalent bond directly onto serine's hook. This paralyzes the body's ability to clear muscle signals. This triggers a cholinergic crisis. Because the signals cannot be cleared, the lungs fill with fluid, and the muscles governing respiration go into terminal spasm. A single chemical lock on one serine residue stops the body's ability to breathe. Whether through the genetic starvation of myelin or the chemical hijacking of an enzyme, disrupting the serine supply chain halts the essential signaling and insulation of the human nervous system. Pathologists detect these anomalies using liquid chromatography tandem mass spectrometry. This system filters blood or spinal fluid particles based on their exact mass-to-charge ratio. It allows the laboratory to quantify serine levels down to nanomolar thresholds, ignoring the millions of other proteins in the sample. To diagnose the starving infant, the pathologist looks for near-zero serine levels in the cerebrospinal fluid. For the paralyzed farmer, they measure red blood cell cholinesterase activity. If it has dropped below 20% of baseline, the enzyme toxin bond is confirmed. But the data is fragile. If red blood cells are mechanically torn by a difficult blood draw, they leak internal serine into the plasma. This creates a false elevation called hemolysis escape, which can mask a life-threatening deficiency. A similar interference occurs if the patient ate a protein-rich meal within 12 hours of testing. This postprandial surge inflates plasma serine levels by exactly 40%, rendering a diagnostic screen for neurological disorders inaccurate. High-resolution mass spectrometry can identify these crises. the result is only as accurate as the physical collection of the blood. If these pre-analytical traps are not managed, the metabolic reality remains hidden. For the infant, the rescue is an act of concentration physics. By administering massive oral serine doses, doctors overwhelm transport systems, forcing the molecule across the blood-brain barrier to restart myelin assembly. The farmer's rescue requires a precise clinical titration. Doctors must push high doses of atropine to block the excess signals. However, they must ignore the patient's constricted pupils and focus exclusively on the lungs, stopping the atropine only when the fluid clears and wheezing resolves. Simultaneously, the drug prolitoxime enters the enzyme pocket. It inserts itself between the serine residue and the nerve agent, using its own localized charge to break the covalent bond. Physically pulls the toxin off the hook and reactivates the enzyme. These interventions work because serine is the backbone of our neurological architecture. It is the mandatory donor of carbon for DNA synthesis, the foundation of the myelin sheath, and the nucleophilic blade that allows our muscles to move and relax. Every memory formed and breath taken depends on the stability of this one reactive hydroxyl group. When that balance shifts, by a genetic error or a chemical bond, the structure of human survival begins to fail.